That's too cute. Aww. We're talking about horses today for those of y'all on Zoom. Okay. Here we go. All right, let's see. Oh, I did not want to do that. Let's see, either one. Okay, we still got a few minutes. I need to do my chat and I need to do the eye clicker. Yeah. Grandad did get hit.
At this rate, we're not going to have a coast anymore.
Well, that's the problem.
And I love that little burrito levy that they made. That was really, they should be real proud of themselves for that. It's called a burrito levy. And I don't know, half of it's already torn out, flying, water's coming.
Here we go. Let's start attendance. Try to get that to go.
No, I've never been, but like, I've heard mixing some people love it, some people think that.
I like it because it's like, it's.
Thank
hands
you.
all by each other. Good on you.
Thank you.
We really like to go back.
I need it today.
But,
Okay. All right.
yeah. So people may have a lot of family and friends, but I don't know.
Okay, so I am going to go ahead and open attendance. Let me minimize. Okay, and if you are absent today, it is okay because I know we got crazy things going on right now. Absolutely. And now let's see if I can write on this. I really, really want to write. We got something on the chat. Okay, Ross. All right. Oh, that's not me. Please tell me this will write. The little things in life. Okay. So we are working on attendance real quick. Give everybody a chance to check in. So Charles and I talked about what joke to share for today. And he suggested we do, what is the richest kind of air? Millionaire. So that is your joke for the day. If anybody has any better ones, I'm always open to better jokes. Thank you, Ross. Okay, so how we doing? Y'all okay? We got a few people in the classroom, which is nice. And Charles is in the classroom with us today. He's going to learn some biochemistry. Or just play on the iPad. Okay, so I will leave attendance run. So if anybody can check in at any point, that's fine with me. All right, so y'all okay on glycolysis? Yes, maybe so. I didn't post my video? Oh, I forgot. Yeah, so there's a song about glycolysis, right? But I also have another biochemistry song that I need to post because Annie posted the glycolysis one, and I need to reply because there's one about, it's for Mother's Day, and it's a biologist who sings about why you're slightly more your mother than your father. And it's all scientifically accurate and hilarious. And so I will post that. Thank you, Annie. I will post that. It's really funny, really funny. Okay, so we're going to go on from glycolysis. And we're going to look at our next step in the process. We're going to look at the citrate cycle. Read it? Saw the video. You saw the funny video? Gotta watch the real videos too. Gotta be a combination there of both, right? So just keep working through it. The trick is to practice glycolysis. Just practice going through it, you know, and once you know what steps are going to happen, you're going to know what the enzymes are, because the enzymes are named by what they do. So once you kind of get a handle on it, it'll get better. Okay, so what's our product? If we have glycolysis, let me write on here. What's our product of glycolysis? Pyruvate. Pyruvate. Now, what can happen to pyruvate? We could go to Acetyl Coa. What else? We can go to ethanol and CO2, right? What else? There's 3 fates. Lactate, right? And so it all depends on the environment around in the cell, right? If we have oxygen present, we're going to convert to acetyl CoA and go into the citrate cycle. So the point of the citrate cycle is to go through some oxidation reduction reactions in order to start gaining energy, right? Because if we go into the citrate cycle, our next step after the citrate cycle is what? Oxidative phosphorylation in the electron transport chain, right? Remember that sort of from what y'all have had that in how many classes now? A lot. Okay, good. Good. I'm glad to hear that. So what you have to kind of remember is that the citrate cycle is not an extension of glycolysis, right? This is the point where we initiate production of energy. So citrate cycle is your initiation. When I get to the top of the screen, it does not like my writing. Our initiation of energy production. E-N-E-R-G, energy production. It's very different. So is glycolysis the only thing that feeds into the citrate cycle? No. What else could come? How else could it come in? Fatty acid synthesis. What else? One more. Proteins. Think about kind of your big food groups, right? Proteins, carbs, fats, right? Those 3. Okay. So it really links a bunch of metabolic pathways, and we'll see that visualized in just a minute. Okay. So this is kind of our outline. Now, when your book does section 1, section 1 is kind of big in the book, but like it's like 3 slides. And it kind of assumes that you have a really great understanding of oxidation reduction reactions. So we're only going to do the 3 slides, but then we're going to work through some oxidation reduction problems and see how it really applies, because I think it's really important. So I know there are only 3 slides, but get yourself like a sheet of paper. We're going to add to it, okay? And then we're going to go into exactly what happens to pyruvate to convert it into acetyl CoA to make it ready for the citrate cycle, right? Because if you've got pyruvate at the end of glycolysis, you can't jump into the citrate cycle. You have to first convert it to acetyl CoA. Then once it's in acetyl CoA, now we can go through the citrate cycle. We'll look at regulation and then what happens with the intermediates within the citrate cycle. Okay. All right. So we kind of talked about this, right? That the citrate cycle can pull substances from carbohydrate metabolism, which is glycolysis, can pull from fatty acid, and can pull from protein or amino acid metabolism. So that's the 3 arrows that you're seeing here. And so we kind of call it the hub of cellular metabolism, because everything kind of feeds into and out of the citrate cycle, right? And we know it's anaerobic metabolism. So what does anaerobic mean? I'm sorry, aerobic. Did I say anaerobic? Aerobic. I'm sorry. I'm sorry. Aerobic with oxygen. Good. And if we don't have this, we're not going to be able to make the ATP that we want in oxidative phosphorylation, right? So you have to go through the citrate cycle in order to get to the point where you're making tons of ATP in oxidative phosphorylation. Okay. And then we said provides metabolism for biosynthetic pathways. So just like you can pull amino acids and you can pull fatty acids into the citrate cycle, you can also go the reverse way. Do you see how these are double-sided arrows, right? So if you're low on amino acids, you can pull from the citrate cycle and use that to metabolize, to create, right? And vice versa, right? We can even pull from fatty acids, right? And then we can convert that to a carbohydrate. We can go gluconeogenesis, go backwards, and increase our blood sugar that way, right? So you have to think about all of these as double-sided arrows. All right, so there are 3 different names for this cycle. So sorry. There's the guy who kind of discovered it, whose name was Krebs. It's also called the tricarboxylic acid cycle. Why do we call it the citrate cycle? Citrate is actually the first product within the citrate cycle. That's why we call it citrate. It's the first product. But it's also called the tricarboxylic acid cycle because guess what? Citrate is a tricarboxylic acid. So citrate is a classification of a molecule, a tricarboxylic acid. Okay. So if you kind of draw back on chapter 2, we can couple oxidation reduction reactions in biochemical pathways to get energy. So we briefly discussed it in chapter 2, but we use that as a source of energy for ATP synthesis. And that occurs, guess what, in the mitochondria. So all of the enzymes that are involved in the citric acid cycle are in either the mitochondrial inner membrane or in the matrix. One of the two. So we've got pyruvate. We're going to have to get it into the mitochondria. So there's actually an antiporter that brings pyruvate into the mitochondria. So it's an antiporter. Antiporter. What is an antiporter? One goes in, the other goes out, right? So pyruvate's going to go in. UV, pyruvate in. And then what you're actually going to have come out is just hydrogen ions. Okay, so once we get pyruvate in, we've got to get it into a form where it can go into the citric acid cycle. And that's right here. That's Acetyl Coa. So do you see how conversion to Acetyl Coa is not really in the citrate cycle? Right? Okay, it's kind of something different. And depending on if you're going from amino acids or fatty acids, where this is going to come in. But this is kind of the basis of a low-carb diet. So what happens if you don't eat a lot of carbohydrates? You're not going to have much glucose. So you're not going to really go through glycolysis. But do you still need energy? Absolutely. So where's your body going to take the energy from? It's going to go first to fatty acids. Now, if you go into starvation mode, you're going to switch to amino acids and you'll actually start breaking down the muscles and stuff inside of your body. But first, we're going to go to fatty acids, right? And so this is how you trick your body into burning the fats that are stored, is that you go low carb, so you can't go through glycolysis, but you still need acetyl CoA, So you take fatty acids and you break them down into acetyl CoA, which can then go through the cycle. All right, so we call this cycle one turn, right? So if you go through the cycle once, you're like going through one turn. How do I do that? So each turn results in a transfer of 8 electrons. And electrons are always going to be transferred in pairs. And free electrons do not exist. They always have to be carried by something. So you'll see half reactions where we say plus an electron, but that's only a half reaction. So you have to remember that there's another half that's going on that's carrying the electrons. And I'll show you some examples of that. You're also going to generate NADH. So you're going to start with NAD plus and you're going to convert to NADH, right? So you go from NAD plus and you're going to generate NADH. You're also going to start with FAD and generate FADH2. 3 NADHs in one cycle, in one like turn around the, I don't know what you call it, but one turn. Then you're going to generate 1 FADH2 and then 1 GDP, which can then be used to phosphorylate ADP into ATP, right? You're generating GTP, and then that extra phosphate group on the GTP can be transferred to an ADP to make ATP, right? So in the end, what we're really trying to make is ATP. But do you make any ATP in the citrate cycle? No, you don't, right? You don't. Look at your overall equation. Are you making any ATP? Nothing. You've got acetyl CoA. You're using up NAD+, you're using up FAD, using GDP, using an inorganic phosphate. That's what's going to turn that into GTP. And you need water. You're going to get out coenzyme A. So this acetyl CoA is an acetyl group attached to the coenzyme A. So when you're done, you're going to end up with just coenzyme A, 2 carbon dioxide molecules as waste, and then your NADH, your FADH2, your GTP, and then you're going to have 3 hydrogen ions. Okay? All right, so it's kind of important to know where you're coming from and where you're going, right? So if you think of the citrate cycle as a metabolic engine, right? Every engine needs fuel. So what's its fuel? Acetyl CoA. You also need O2. If you don't have O2, it's not going to go, but neither does your car. Your car won't go if there's no O2, but we don't say that's the car's fuel, right? Same sort of deal, but know that it's really important. You have to have it. What do you kick out? What do you want to get rid of? CO2. So CO2 is our exhaust. Now, what's the work? Like our car goes somewhere, but our cycle doesn't go anywhere. What does it do? It transfers electrons. Okay, so I guess our goal is through THROUGH, through the transfer of electrons what are we going to do? Yes, transfer of electrons. We have redox reactions. Wes? What is she waiting for? Through redox reactions, right? Okay, so what are our key features? What do we want to take away from this like big overall picture, right? Well, we know it's cyclic, right? It's not a linear pathway. Glycolysis was linear. Citrate is going to be circular, right? So that means if it's circular, that the substrate for the first reaction is going to have to be the product of the last reaction, right? That's the definition of cyclic. So we have to regenerate our products. So oxaloacetate, 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 is going to be our substrate for first and product of last. And you remember how inside of glycolysis we had particular enzymes that regulated certain steps within glycolysis in order to maintain control? There were 3 of them. You should really know what those are. Hint, hint, hint. You should really know what those are. Within the citrate cycle, it's different. There's nothing inside the citrate cycle that really regulates it. It's going to be regulated above when you're converting into acetyl CoA. So our regulation for citrate comes before the cycle, not during the cycle. Okay? Okay. So I think, see, yeah, okay. So your book kind of like says, oh, that's enough on section 1, but I really don't think that that's a good idea. So so take out your sheet of paper, right? There are 2 enzymes, that regulate citrate, citrate cycle, right? So those 2 are pyruvate. Got to think about this. Pyruvate, we got to convert pyruvate to acetyl CoA, right? So we're going to have a pyruvate dehydrogenase. And that dehydrogenase should be a light bulb for you that there's going to be an oxidation reduction reaction going on. So in organic, did y'all do that in organic? Like oxidation reduction with carbons?
It's different like reactions. Like you had a catalyst and like that.
Okay, okay, that's okay. I kind of figured that, so we're gonna kind of start from scratch. So it's all good. It's all good. I'm gonna show you because we have to get pyruvate to acetyl CoA, and that's one of the steps inside of it. Yeah, yeah. So we're gonna look at the structure. I promise we're gonna look at everything. I promise. I'm not gonna skip any steps. Oh, I will not. I will not. All right, and then pyruvate carboxylase is our second one. These are our 2 enzymes. Are these enzymes in the citrate cycle? No. So make sure you remember that. Not in citrate cycle. Okay, so let's do a little bit of a review on oxidation reduction. Okay. It can. Yes. 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 Okay, so redox reactions. Let's start with this. When you think redox reactions, we did this in class before. What's the little shorthand that you should remember for oxidation reduction reactions? Oil rig, right? So oxidation is loss of electrons. Reduction is gain of electrons. But I said there was something else you had to worry about when we were talking about organics. So oxidation is gain of oxygen, loss of hydrogen. So look at this, dehydrogenase. What do you think? So now we know we're talking about oxidation reduction. Oh, awesome. Okay, so reduction is gain of hydrogen, loss of oxygen. Okay. So what we're going to be doing in redox reactions is really transferring electron pairs. Where are they coming from? from some kind of an organic molecule, right, into a carrier. So why? Why do they move? Why do electrons move? Well, like if I were to ask you why is water, yes, Adela's got an electron affinity difference. Why is water polar? Because oxygen is more electronegative than hydrogen, right? And so that's exactly what's going to happen in these redox reactions. Exactly the same thing, right? And so this exchange or this transfer is due to electronegativity differences. Which change our electron affinities. So if the two compounds, the organic molecule and the carrier, have the same electron affinities, it's not going to happen. So what you need to have is start with something that has a really, really low electron affinity and transition to molecules that have stronger and stronger and stronger electron affinities until you get to the maximum electron affinity. Anybody know what that is? Huh? No, what molecule? What molecule has the highest electron affinity? Think almost water, but not quite water. That's the atom, but I'm talking about a molecule. Yes, fluorine. Y'all know what I call fluorine, right? No, fluorine the floozy, because she's so electronegative that she'll take electrons from anybody. She's fluorine the floozy. That's how I remember, you know, and whoever's closer to fluorine is more electronegative. O2. O2 is our maximum electron affinity. So that's going to be our end game. That's where we want to get our electrons to at the end. And so when we transfer those electrons to O2, what we end up making is water. And that's why water's an end product of all of this. So no, but in this case, they're going to. Just in this case. Just in this case. Okay. So you have to know a little bit of vocabulary because they're going to throw it at you and expect you to know it. So I want us to kind of review it. So if you are an oxidant, what is an oxidant? A compound that accepts electrons. And these become reduced. Okay. So what do you think? Reductants. Yes, compounds that donate and become oxidized. Great. Okay. All right, so we usually have that gain or loss of electrons at carbons, right? We're talking organic molecules here, so you got to kind of think about carbons. But I'm going to use some examples that aren't carbon because they're really good illustrators, and then we're going to use them in the oxidative phosphorylation stage. So you'll see them again. So if I have Fe2+, what's going to happen to Fe2+? It's going to gain an electron, and it's going to go to Fe3+. Do we have free electrons floating around? No, no, but this is only a half reaction, right? So our Fe2+ and our Fe3+, these are our conjugate pairs. Remember when you did acid and bases, right? An acid turns into a conjugate base, a base turns into a conjugate acid. Same thing, same thing. So what about Cu plus? Cu2 plus what? Whoa, not a big 2. Cu2 plus, and then what? Plus an electron. Wouldn't an electron make it lose charge? Oh, sorry. Oh, sorry. So let's go, usually we do it, I'm sorry, I should have done it this way. Fe3 plus, Fe2 plus. This is kind of, it doesn't matter which way you draw it because they're actual reversible reactions, so they can go either way. So yes, it should be 3 plus going to 2 plus. Okay, so what we do inside of the cell is that we couple these reactions, right? Remember how we talked about coupling ATP, right? Same thing, we're going to couple oxidation reactions, all right? So what happens if I couple these two? What do you notice? Well, I have an electron on this side, and I have an electron on this side. So what's my overall reaction if I wrote this out? Fe3 plus plus Cu plus goes to Cu2 plus plus Fe2 plus. Do I write my electrons? They cancel out. What do we call those? We did this before. Anybody at home? Not spectators. Intermediates, yay, intermediates. So remember how this will drive reactions, right? Same sort of deal, same sort of deal. So in the first one, what are we doing to iron? Are we oxidizing or reducing? We are gaining electrons, right? Always write your oil rig. So we are reducing Fe. Can I move this? It's like too many little things on the screen. All right, and so what are we doing to copper? We're oxidizing, right? So there are enzymes that catalyze redox reactions that have iron and copper in them in the electron transport chain. So we're going to actually see this in use in the next chapter, right? But let's talk about the citrate cycle. It's easy to start talking about metals because y'all have seen that before, right? Good. So now we're going to transition this into organics, right? All right, so in the citrate cycle, we have redox reactions that involve the electron pairs being transferred from NAD plus, NAD plus, and what was our other one? FAD. All right, these are our important ones. So let's talk about NAD. Let's see if I can go down, okay? So if we have NAD, and we add a hydrogen ion. Oh. And y'all, have y'all ever seen this before? Yes? I see some yeses, I see some no's. Okay, all right, so it's not a big deal. So in this one, and let's write this out, it goes NA, I write too fast and it can't keep up. It just gives me little dashes, right? NAD plus H plus. So our reductant is going to give up 2 hydrogen atoms. One as a hydride ion. This is a hydride ion. So if we have a hydride ion, okay, we have 2 electrons, that's what the 2 little dots are, and we have 1 proton. So what's my overall charge going to be? -1. So that's why we have a negative sign, right? So that's a hydride ion. And then this we call a proton. Why do we call it a proton? It's just a proton, right? If you look in the periodic table, what's the atomic mass of hydrogen? 1.008, right? So why isn't it 2? Don't they have a proton and a neutron? No, the most common form of hydrogen, very, very common form of hydrogen, doesn't have a neutron. It only has a proton. Anybody ever thought about that before? Yeah, so it only has a proton, the most common one, right? Okay, and then because it doesn't have any electrons, because it's an ion, right, it's positive. Okay? All right, so what's going to happen if we remember our oil rig? Let's do oil rig. What's happening in this? Tell me, using our oxidation and reduction terms, what's going on? NAD is gaining electrons. So another way to write this would be NAD plus 2E minus, right? And what else are we gaining? What do we have over here? 2 hydrogens. So you could write it this way, right? Yields NADH. This is probably how you've seen it, plus H plus. That's probably how you've seen it in the past. Yeah? No? But y'all have done? No. Never seen that. Okay. All right. Well, no, this is good, important information to know. I want y'all to tell me. I absolutely want y'all to tell me. Uh-huh. You're going to pick up one of the hydrogens, and the other hydrogen is going to be free. And then you're also, as NAD plus, going to pick up 2 electrons. So now this is the electron carrier. So if we look at what's oxidation, oxidation is loss of electrons. Reduction is gain. So NAD is undergoing what? It's undergoing reduction. So it is the oxidant. I know, right? So if you come up here, oxidant, compound that accepts electrons and becomes reduced, right? I know this gets confusing, and I really don't like that they do this, but they're going to use it throughout the entire book. Accept and gain. Yeah, accept and gain. Same thing. Same thing. Yes. Yes. All right. Okay, so if y'all have not seen that before, let's do the same thing with our other carrier, FAD. Okay? So what's going to happen with FAD? So FAD actually occurs in 2 reactions. Whereas NAD was 1 reaction, FAD is going to be 2 reactions. So just this molecule can only accept 1 electron at a time. Okay, so FAD plus 1 electron plus 1 hydrogen ion is going to give you FAD. And then this is a temporary molecule, and so you'll see kind of a dot right there indicating that it's only 1 electron, right? And it's very unstable and doesn't stick around for very long. So we have our 1 electron, and then we're just going to give it another electron, but we also need another hydrogen. I guess I didn't really need to write the 1, right? You could just write it alone. That might make it easier. Plus H plus goes to FADH2. And all of these are actually reversible. I don't think I wrote that on the other ones. They're actually reversible reactions. So here FAD is being reduced, right? So it goes through one reduction and then it goes through a second reduction to end up with our fully reduced FADH2. Yeah? Not too bad? What's that? These are still half reactions. Yes. If you ever see electrons written in an equation, it's a half reaction. So it's like 2 half reactions. So they're like 1/4 reactions.
So for the other one, the N
Uh-huh. This
even
is,
though we have 2 electrons, it's still a half.
it's still a half reaction. Anytime you see free electrons, it's a half reaction. A half reaction is just telling you you're only looking at one species. Because as one species is being reduced, another species has to be oxidized. And so usually it's one of them is the substrate, like the carbon containing molecule, and then one's the carrier, right? Okay, so in the very end, right, what we want is for oxidation to transfer those electrons to oxygen, right? Oxygen wants to carry those electrons because of a difference in electronegativity, right? So you're usually going to be looking at electrons moving from carbons to oxygens, C to O, because oxygen is more electronegative. So if you're looking at a pure oxidation reduction reaction, you're going to call it an oxidoreductase. That's the enzyme that's going to catalyze it, right? But typically in organic, we have a loss of one or more of hydrogens that go along with that oxidation, right? And so what are we going to call those enzymes in organic? Organic, we usually, usually, usually we have a loss, right, of hydrogens. So what are we going to call that? What enzyme is going to catalyze a loss of hydrogens? A dehydrogenase. Great, yes. So did we see a dehydrogenase listed there? I believe we did. Right? So we have two primary energy conversion reactions in metabolism, right? The one we talked about, so 2 primary energy conversion. So the first one we talked about in glycolysis. How did we get energy in glycolysis, right? We did make some energy in glycolysis, didn't we? Our ATP, exactly. So phosphoryl, P-H, oh my gosh, phos, P-H, I see, I have a letter that's wrong and it drives me crazy, H-O-S-P-O-R-Y-L, phosphoryl, transfers involving ATP. Now, what's our second one that we just learned? Oxidation reduction, right? So these are going to use electron carriers. What are our electron carriers? Now you know they are NAD plus going to NADH, and what's my other one? FAD going to FADH, 2. It doesn't. It doesn't. It'll go reverse. It could go reverse because all the reactions are reversible. So it'll just go backwards. but it won't be stuck. It's like you're never stuck at that transition state. Same sort of deal. Yeah. Okay. So before, we've always talked about delta G on if a reaction is going to go or not, right? But with redox, we have to worry. Yes, we have to worry about delta G, but what does delta G depend upon? In this case, we're talking about a reduction potential. Okay, so if you remember back to, I don't know which chem, y'all will have to tell me, but your biochemical standard reduction potential. Did y'all do this before? E as E? Y'all do volts? Y'all do any of that?
I think that was
Chem 110. Yeah, it's probably been a while. Probably been a while. Yeah, that's all right. So this just represents electron affinity, right? We're not going to get into the super complex details, but it basically represents electron affinity. And we always compare it to a hydrogen half reaction. So what's a hydrogen half reaction? There we go. 2H plus 2E minus. Goes to H2. We basically set this at 0 for kind of arbitrary reasons, a little bit, right? But this standard reduction potential, remember when we talked about standard conditions, right? 1 mole of substrate in product, pH 7, 25 degrees Celsius, right? It's all the same. So all of those conditions are exactly the same. And so when we compare the standard reduction potential of NAD+, of FAD, all that kind of stuff, we're always comparing to this hydrogen half reaction, right? And so if you have a standard reduction potential, it's like this, not like that, that has a higher affinity than H+, then this is going to be positive. So what if you have one that's negative? What if you have a negative? That just tells you that this standard reduction potential is lower affinity than H+. So it's how much it wants those electrons. Remember how we talked about positive delta G, negative delta G, right? It's kind of the opposite. It's kind of the opposite. Yeah. Okay, so you always look at coupled reactions because that's how they happen in the cell, right? We talked about coupled reactions with ATP. We're assuming, yes, we set, Taylor, yes, we set hydrogen's affinity at 0. Yes. So we're always going to talk about coupled reactions because that's what goes on in the cell. Just like we talk about coupled reactions in terms of delta G, we do the same thing with our energy that's available, our reduction potential. So we're always going to express that in terms of our acceptor, right? You can put electron acceptor if you want, minus our electron donor, right? Because it's coupled. So you have to look at both the acceptor and the donor. And this is what controls our free energy for the cell. So have y'all seen this equation before? You're not going to have to plug anything into it, but it's just important to see so you understand how it relates to delta G. No? This is not one? Y'all didn't do any electrochemistry? I taught electrochemistry at the high school level. I think my kids got a really good challenge. Like, What's that?
Depends on the compressor, what's heat temperature.
But there should be a common base.
They don't want to like temperature stuff.
Yeah. Okay. So this is an equation. Oh, why did I do that? It's G. No wonder y'all were like, I don't never seen that. It's G. Y'all still no. Are you going to post it? Of course I'm going to post the recording. Yes, of course. Of course. So do you notice that there's a negative sign here? So that means that if this is positive, delta G will be negative. If this is negative, a negative and a negative make a positive. That's why. Like, I feel like it's just important to expose you to it so that you understand why it's opposite. Okay, so this is Faraday's constant. This is our like number of electrons, Faraday's constant. I don't even know how to spell Faraday's. F-A-R-A-D-E-Y? A-Y? Whatever. Faraday's constant. Did not get a PhD in spelling. And so the number of electrons is never going to be negative, right? Faraday's constant is a constant, it's positive, it's never going to be negative, right? So what's going to control the sign of delta G? Our reduction potential. Exactly. Okay. So if you have, in order for this like chain, it's going to be a sequence, it's going to be a chain of events that we're going to follow, right? And so when you have differences in our reduction potentials, and coupled reactions, coupled redox reactions, right? What do we want to happen? What has to happen so that those electrons keep jumping from one to the next, to the next, to the next? Our reduction potential for the acceptor must be what? Positive. Must be more, we'll just say more positive than the donor, right? Otherwise, the electrons won't move. Does that make sense? This is why electrons are going to move. And I think it's super important that you understand this before we try to get into the nitpicky details of what happens in the citrate cycle. So what did I say was the most potent oxidant? Whoa, yes, most potent oxidant equal to O2. So do you want O2 at the beginning of all this, or you want it at the end? We want it at the end. And then O2 is going to pick up those electrons and turn into water, and that's why water is one of our products. So Ross, you should definitely go and read the book. That's going to really help. That's going to really, really help. Now, I want to tell you, on the last page of, I'm about to end, but on the last page of this section, I don't remember what page it is. Last page of this section. So last page of section, this is 10.1. There's this little question, right? And it asks you how reduction potential is related to metabolic pathways. I want you to cover the answer and try to answer it for yourself. See what you come up with and compare it to their answer and start to try to think through this. Because it's a really good question. And it helps to make sure that you've integrated all the information from all of this. Okay. Now your book goes into a little more detail in the standard reduction potentials, but you don't need that. You just need to understand how it works. Okay. So we'll pick up on section 2 the next time I see you on Monday. Okay. Let me know if y'all need anything, if I can help you in any way. But I'm so glad that I decided to go into this. I think this is a good decision. So you don't have videos to watch. No, you do not. No videos to watch. Go read the book to make sure that you're ready for lecture on Monday. Read the book. Okay? Any other questions on Zoom? Everybody okay? So go practice that question at the end. It's a really great pull it all together question. Sometimes I like them and sometimes I don't. This one I really did. Okay. Y'all have a great weekend. I hope everybody gets power back. And please let me know if you need anything at all. Saying I need to read the book and do any other kinds of practice. Like you could reading the book would be really good. I would suggest go back and reread section 1 and then read section 2 and possibly even section 3 to get ready for Monday. Because I don't know that section 2 is going to take us the whole time. I think we'll probably get into section 3 as well. Okay, Ross? Great. All right, y'all have a wonderful, wonderful weekend. I'm going to go ahead and end the Zoom if anybody has, unless anybody has questions. Okay. No questions? All right. Talk to y'all later.